Simultaneously, the centrioles what are present over here. So a pair of centrioles are lying right angles to each other, and these centrioles start to move apart. Means both of themselves they start to move apart. Here one centriole that comes over here, and one centriole that remains over here. And so these centrioles, when they start to move, so towards both the poles, so this is one pole and this is another pole. Simultaneously, they start to produce minute microtubules. Minute microtubules start to arise from these centrioles, and they are looking to be as stars. Hence, these are forming the asters. So, what they form? Asters. So, they start to radiate small microtubules from themselves, and both of them start to move apart. So, towards both the poles. One remains here, another one start to move over here. So here what happens? So as a result of the radiation of these microtubules from these centrioles, so they look to be as stars, hence they are called as asters. Means here centrioles start to move towards the opposite poles. They start to move towards the opposite poles. Okay, they start to move towards the opposite pole, and simultaneously, what happens over here? So they start to form the spindle fibers. Right? Initially, the spindle fibers are started to form over there. And this nucleolus gradually start to become smaller enough. Right? It start to become nucleolus. Start to become smaller enough. And gradually what happens? Nuclear membrane also start to disintegrate gradually. It start to disintegrate. See here, it start to disintegrate. Right, like this. It start to disintegrate like this. Okay. Disintegration of nuclear membrane is taking place. Gradually the formation or the condensation of this chromatid into chromosomes. So chromatin into chromosomes that is taking place. Gradual disappearance of the nucleolus is taking place. Movement of the esters towards opposite poles is taking place. All these are the activities that take place over here initially. And at last, post-prophase it is said as. This is so prophase initially what takes place. Post-prophase, in post-prophase what happens? Disappearance of nuclear membrane and nucleolus takes place. This leads to leaving the chromosomes directly into cytoplasm. See here, now the chromosomes they came into the cytoplasm. Why? Because the gradual disappearance, complete disappearance almost. See here, disappearance of both of these. Now where the chromosomes are? They are in cytoplasm. Right? So this is the fact what happens in case of the prophase. Okay? So in prophase, this is the activity that is done by the nucleus itself. Simultaneously there the cell organelle is like endoplasmic reticulum, Golgi complex, they become inconspicuous, right? Simultaneously, the Golgi complex and endoplasmic reticulum become inconspicuous. So very negligible. So inconspicuous they become, and simultaneously, so the esters what they form the spindle fibers, it is taking place only in case of the animal cell, not in case of plant cell. Why? Because esters are absent in plant cells, centrioles are absent in plant cells, hence the spindle formed is an estral in case of plant cell and it is amphiestral in case of animal cell. Right? Point to be noted. Amphiestral in animal cell and anestral in plant cell. Alright? The spindle fiber is amphiestral in animal cell. Why? Because two esters are formed over here, from which the esters are formed over here, from which the spindle fiber is formed. But so anestral in the sense that there the spindle fiber is formed without centrioles. Next stage is metaphase. In metaphase, what happens? It is a short period stage, 2 to 10 minutes it takes, right? It takes 2 to 10 minutes over here. In metaphase, what happens? Let us see here. This is the cell. So where the nuclear membrane is totally absent there. So the esters, they have become to the poles respectively, and these have formed already the spindle fibers over here. Okay. So here what happens over here is, if the chromosomes are there, they are start to coming towards the equatorial line. This is the line called as equatorial line. So all the chromosomes are brought to the equatorial line with the help of these spindle fibers. With the help of these spindle fibers. How they are brought to the equatorial line is the mat over here. Okay. So they are lying just like this. This is the centromere of one chromosome. So these are the chromatids of the chromosome here. So they are attached. They are lying on the equatorial line like this. A single diagram I have drawn for your convenience purpose. Okay. What happens here? So across this cell, all the chromosomes are lying initially. So in the post-prophase stage, in the pre-metaphase stage, what happens here is, so the attachment of these chromosomes to the spindle fibers take place. Before that one, the formation of this spindle apparatus is formed. Here, formation of spindle apparatus has completed. After this one, after its completion, what happens here? Spindle apparatus is formed over here, and chromosomes attached to spindle fibers, right? How do they attach to the spindle fibers? So whenever you come across one chromosome, you know this is one chromosome to which a centromere is present, So chromatids, these are chromatids. Chromatids are attached to the centromere, right? It is also called as primary constriction, to which C-shaped, disc-shaped structure is so attached on the centromere, and it is called as kinetochore. Kinetochore, we know it. So from these one, there you come across this kind of fibers or the microtubules. These fibers are called as chromosomal fibers. What these are? Chromosomal fibers. These chromosomal fibers are attached to the spindle fibers. This is how the chromosomes attach to spindle fibers with the help of chromosomal fibers arise from kinetochore, right? So this is how the chromosomes are hanged to the spindle fibers formed there. Okay, now we came to know how the attachment of these chromosomes that take place to the spindle fiber, and simultaneously these chromosomes are brought towards the equatorial line, right? See here, chromosomes are brought to the equatorial line, and our line on equatorial line in such a way that the centromeres of all chromosomes lie right angles to the spindle fibers. See here, the chromosomes are lying just like this, right? And one more chromosome I shall draw over here. See, this is also the centromere of one chromosome. It is lying like this. Okay, this is one chromosome. Now we came to know these chromosomes are hanged to the spindle fibers, and they attach to the spindle fibers, and they are brought towards the equatorial line. So what it looks to be as is, this is another centromere, this is another centromere, this is another centromere, this is another centromere. See here, all the centromeres of the chromosomes are attached to the spindle fibers here like this. These are all spindle fibers. So to which the attachment of chromosomes that take place over here. Like this, all are lying in the equatorial line. So that what happens? All chromosomes lying on equatorial line to form a plate-like structure called as equatorial plate, right? Equatorial plate is called as. So that kind of structure it forms over here. In this case, metaphase chromosomes are lying on the equatorial line formation of equatorial plate that take place over here. So this is one. And there the fibers, already I told you there are three types of fibers that we come across over here. What kind of fibers these are? One is continuous spindle fibers, one is continuous spindle fibers, and second one is discontinuous spindle fibers, and third one is chromosomal fibers. Chromosomal fibers. Continuous fibers, discontinuous fibers, as well as chromosomal fibers are the three kinds of fibers that we come across here. See here, so these are the fibers that we come across here is, this is one cell, this is one ester, this is another ester. See, they are continuing from one pole to another pole, right? If they continue from one pole to another pole, then they are called as continuous fibers. They are called as continuous fibers. Sometimes what happens, some of the fibers which are making the discontinuous one, they come on here and stop themselves. They do not reach another pole, right? They do not reach another pole. They are called as discontinuous fibers, right? One. And chromosomal fibers, you know it better. From the kinetochore, whatever the fibers that arise, and they hang to the, or they reach to the one pole of the cell, then it is said to be as, chromosomal fibers. That kind of three fibers that we come across over here. And last point and very important point, the process of bringing all the chromosomes on the equatorial line, forming an equatorial plate, is called as congregation. Right? What do you mean by congregation? It is nothing but bringing of all the chromosomes on the equatorial plate. So it is done by the spindle fiber. So this is called as what? So the metaphase. So these chromosomes are highly thickened one, highly thicked, darkly stained, right? These are the chromosomes which are suitable for their study itself. Right? So this is one more fact to be remembered regarding the metaphase. And the next stage is anaphase. In case of anaphase, what happens? Let us see. Next stage is anaphase. It is a rapid stage of cell division in which movement of chromosomes take place. Movement of chromosomes take place towards opposite poles of the cell. See a rapid movement of the chromosomes that take place on the opposite poles that occurs in case of anaphase over here. See, what happens here is, already the chromosomes are hanged to the spindle fibers. This is one ester, this is another ester, one spindle fiber, this is another spindle fiber, to which the chromosomes are hanged over here, right? Now all are lying in the equatorial line. I am going to show only two chromosomes over here. This is one chromosome with two arms. This is another chromosome with two arms. See, whenever these are lying in the equatorial line, in case of anaphase, where the breakdown of this centromere that take place. This is the centromere, the breakdown of this centromere that take place. See here, this is one chromatid, this is another chromatid, this is chromatids like this. So now what happens over here is, so the separation of this chromatid, this part of the chromosome, this part of the chromosome, that takes place due to dissolution of the cohesin protein which attaches these two chromatids to the centromere. When it dissolves, both of them split. this one becomes separate chromosome and this one becomes separate chromosome. And these chromosomes are said to be as daughter chromosomes and they are said to be as monad. Monad in the sense that chromosome with only one arm, right? See here, this kind of chromosomes are moving apart each other. This part of the chromosome is moving this way and this part is moving this way. Why? Because these are attached to separate spindle fibers which are continuous spindle fibers. Sometimes they are attached to the spindle fibers along with chromosomal fibers they are attached. So these chromosomal fibers are also there as well as spindle fibers are also there to which the chromosomes are attached. Now what happens? The chromosomes are pulled apart. When the chromosomes are pulled apart with the help of the spindle fibers, especially chromosomal fibers which are arising from the kinetochore itself, there are some protein motors are there, and those protein motors they help in sliding movement of these chromosomes. They help in sliding movement of chromosomes with the help of dynein and use of ATP molecule. Loss of ATP molecule take place and dynein protein is there. It helps in the movement of these or pulling apart these chromatids or chromosomes towards the opposite pole. Is it clear? Now, see here. So this chromosome it start to move towards this pole and the centromere is at the forward and the chromatids are following the centromere. Why? Because this is the region where the kinetochore are, so the fibers are there and those fibers are called as what? Chromosomal fibers. So they start to what? So there due to one of the reasons they start to move. One reason is there the proteins are specially present in the kinetochore part, and those proteins I have already mentioned. Motor proteins are there, right? They are dyneins and they transport these chromosomes towards the pole, right? So there are proteins are there, they are called as motor proteins. These motor proteins, dynein is there with the use of the ATP. These protein help in the movement of these chromosomes. One thing, and simultaneously disassembly of chromosomal fibers also take place. When the disassembly of chromosomal fibers that take place, that moment what happens, shortening of these microtubules that take place, that is nothing but kinetochore fibers that take place. Whenever they become short, that moment these start to move towards the pole. Imagine every fact over here. Why? Because it is not shown over here. So due to all these reasons, the chromosomal movement that take place. That's why we draw the diagram like this in anaphase. So if it is equatorial line, this is one pole, this is another pole. In case of animal cell, spindle fibers are formed like this. These are continuous fibers to which the chromosomes are hanged over here. See here, this is the centromere, this is the centromere, this is the centromere. It is seen in case of metaphase, but the centromeres they come over here, to which the chromatids are attached. See here. chromosomes are moving in this direction simultaneously. Other chromosomes are moving this direction. So these are moving this direction, these are moving this direction. They are moving totally towards the opposite poles. Right? So this is how the rapid movement of the chromosomes that take place in anaphase. And the next stage is the telophase, and it is the last phase of the karyokinesis. What happens there in telophase? Let us see now. Next one is telophase. Telophase. It is a long period stage during which what happens, ultimately the chromosomes that reach towards the pole. Okay, so see here, this is the animal cell and the chromosomes that reach the poles over here. The chromosomes that reach the poles over here. This is one chromosome, another chromosome. So totally these spindle fibers are also there itself. Ultimately the chromosomes reach the poles, and these chromosomes they start to uncoil themselves. The condensed thread-like structures, that is chromosomes, they start to what? Uncoil. Chromosomes start to uncoil themselves, forming chromatin network. Okay, these are all chromosomes which are present in the polar sites. Okay, they start to uncoil to form the chromatin network, in the sense that this is the cell in which the chromosomes start to uncoil, forming the chromatin network itself. Chromatin network simultaneously. So the spindle fiber start to disappear and start to come over here at the central axis of the cell. Okay, and simultaneously the reappearance of the nuclear membrane starts. The nuclear membrane that starts to reappear, and simultaneously the nucleolus also start to form. Nucleolus also start to form. How this nucleolus is forming? With the help of what kind of chromosomes, you know? So with the help of the SAT chromosomes, right? So these SAT chromosomes are usually present in 5 pairs in case of human being. Already I have mentioned the names also, 13, 14, 15, 21 as well as 22. So these SAT chromosomes have nuclear organizer region is there. With the help of this one, the nucleolus is found over here, and after that one, after the reappearance of this nuclear membrane as well as the nucleolus, what happens? 2 separate nuclei are respectively formed in a cell. 2 separate nuclei or daughter nuclei are formed in the cell, single cell itself. And this cell undergo what? Next stage, that is cytokinesis. This leads to cytokinesis, means cellular division itself, right? So cell division, how it occurs in case of plant cell as well as animal cell? This is the question. Okay, so This is all about nuclear division. This is all about nuclear division, means 4 stages that we have explained over here. One is prophase, second one is metaphase, anaphase as well as telophase. These are the 4 phases that we come across over here. So this is all about what? The nuclear division. Next one is, that is cytokinesis. Right? So next one is cytokinesis. How this cytokinesis that occurs, so in plant cell as well as animal cell, let us see now.